the new technology like tissue culture, and in tissue culture how we are using that organic farming, because even the lay person now we are able to understand about the organic concept. So even when you go to the market, affordability is little bit high for organic vegetables, but the people are buying and using it, and more awareness is needed to the general public about the organic. And also the topic, which is a very scientific topic, which was selected by the HOD today. I think this will be very useful for the students. And once again, I like to thank the organizer for giving the opportunity to share my views regarding this Cassava biotechnology. Thank you very much.
Thank you, sir. Now I request our speaker, Dr. N. Narayanan, sir, to deliver his talk.
Thank you so much. Can you all hear me? Can you all hear me, right?
Yes, I can hear, sir.
Yes, okay, sir. Very good. Okay, thank you so much. I would like to thank Dr. Muralidharan and Dr. Santosh Babu and like my friend Dr. Rajarajan for inviting me to give this lecture, and I'm really excited to present to really present my work on Cassava biotechnology. So let me share my slides. Can you see my screen? Can you all see my screen, right?
Yes, sir. Yes, sir.
Oh, okay. Very good. Okay. Okay. So, today I'm going to talk on Cassava biotechnology, applications, and challenges. So before that, I just wanted to give what are the problems that we are facing right now in the world, and what are the projections of it. Like, for example, the world population is expected to reach 10 billion, and the food production should increase by 70%, and the energy use is predicted to be double. So we have a lot of problem that is coming through to us. So with all this in mind, so we have a lot of challenges that we are projecting in 2050s, and with that in mind, our center is called as the Donald Danforth Plant Science Center, which is at the St. Louis region in the Midwest region of the United States of America. Our mission of the Danforth Plant Science Center is to improve the human condition through plant science. I know medical science is doing its work, but at the same time, in order to improve the agriculture, we can also improve the human condition through the plant science. How are we going to do that? So we can feed the hungry and improve the human health. We can preserve and renew the environment. And the third objective is to enhance our own region as a world center for plant science. And we are collaborating with all international scientists. In that way, we can partner with them and then present our work, like what we are doing right now. And just one or two slides about our own center. A Danforth Plant Science Center is a non-profit organization, and you see the building in the top here. And we have around 300 employees, 25 scientific teams, and people are all over the world. We have around 24 to 25 countries are participated in our center, and we turn around $200 million in research funding from the government and agencies and foundations.
Sir, Narayan sir, sorry to disturb. Full screen, sir.
Oh yeah, I will try to do that. If I do the full screen, what happens? So let me try that. Okay, can you see now?
No, sir.
Can you see the full screen or no?
No, sir.
Okay, let me do that because the full screen doesn't work. Let
Okay,
me
okay,
do
no
that.
problem, sir. Okay.
Okay, let me do that. Just a minute. Okay, can you see my screen now
Sir,
again?
PPT mode, sir, PPT mode.
I did that, but when I do that, it doesn't work because I have 2 or 3 monitors here. So because of that, it doesn't work. So can you see this slide, the one that I'm talking about, the Institute for International Crop Improvement?
Yes, sir. Yes, sir. Yes, sir.
Okay, okay, good. Maybe I will just reduce this a little bit. Okay, okay. Yeah, so I was talking about the Danforth Plant Science Center. So just moving on to my own topic and research, that what I am passionate about is like we all do a lot of like work and like inside the lab, but our major focus and my passion towards is like to turning the discoveries that we do in the lab, take that into the field, take that food and the nutrition for the nutrition security. So how are we going to do that? I'm just going to show a couple of examples at what we did. So in order to do that, we have to do the product development, field testing, and then like regulatory and stewardship, and then designing deployment systems and capacity building. So I'll just explain more about that. So like kind of like so like whatever that we are doing like basic research in the lab and the greenhouse, that will move on to proof into the field trials. So it will go into the field, and then it will go for the performance and regulatory trials. Like in all the stages of this, we will be training the people, and that will go into a certification, and that goes into the farmers field. So that is what is our own impact, that how we are going to achieve this impact in Africa. So like we are like working on cassava. So I'm just giving an example of African country. So I'll talk about like what is cassava. Like if some people, some students have not heard about cassava, I can talk about that. So I'm going to show 2 examples in this presentation. So the first one I'm going to talk about is on the disease resistance, like what Dr. Santosh was telling about the pest and disease resistance. And the second is about the like biofortification, which is like the nutrition part. So first, let me introduce the crop, cassava. Cassava is one of the most important crop in Africa, and it is consumed by more than 800 million people in Africa. And it is so this is a storage route. I'm sure in India we have cassava. In Kerala, I know that Kerala people eat a lot of cassava. So and also so these are the stem cuttings, and stem cuttings is the way that the cassava is like propagated. So I know in Kerala, like they do a lot of research on cassava. There is a specific institute for that. We have talked with them also, and this is a field trial institute of the cassava. So this is a major component of food and economic security. It is called as a poor man's crop because everyone take cassava stem cuttings and put it on the ground. So like it can be banked inside the store, like inside the soil for 2 to 3 years. So when people don't have money to go and buy food, they go and take the stem cuttings of cassava and they propagate and they take the food from cassava. So that is why it's called poor man's crop. So we want to feed the entire nation. We want to feed the entire world. So that is why like this cassava is one of the most important crop. So the kind of the problem that I was talking about is like 2 diseases. So one is the cassava mosaic disease, which is caused by a Gemini virus DNA, like virus. So both are virus, like so diseases. So the first one is the cassava mosaic disease, which is like which we call as ACMV and EACMV, which is African cassava mosaic virus. And there is another one, it is called as the cassava brown streak disease, which is an RNA hypomoviruses. And this is called CBSV because it is called cassava brown streak virus. And economic loss due to cassava is up to 100% and spreading rapidly throughout East Africa. So from our center, we have been working on cassava for the last 15 years. And so we have developed several technologies. So I'm going to show one of the technology what we have developed. So we have developed an RNAi construct. RNAi, so the construct is kind of like having two pieces of the code protein of the gene, which has an intron in between. So it is an RNAi technology. So you will have that, and that is coded into a binary vector, and that binary vector is transformed into agrobacterium, and then we will then take it into the plant. I'll show that the procedure in the next slide. So we have identified a very famous cultivar that is in Uganda called as a TME204. So it is highly susceptible to cassava brown streak disease. So with various studies that what we have done in 10 years, we have developed this piece, which has the two code proteins of the virus, which is like intermediate with an intron. And this piece is inserted into agrobacterium, and then it is transformed into the plant. So I remember just like few minutes back, Dr. Santosh was talking about tissue culture, and that is the same way that we have been developing this. So we start with a small leaf explant, and then we go into embryogenic structures called as a friable embryogenic callus, and then we do an agrobacterium transformation, and then we have antibiotic selection, and then we get a transgenic plant, and that plant is going through a lot of molecular screens, tDNA, copy number, vector backbone, gene expression. So we do a lot of screening, and then we do a mass propagation of those plants. From St. Louis, we will be shipping, and then we will do a phenotyping testing in our own greenhouses. And then after that, it goes into a confined field trials in Africa. So we ship all these plants from St. Louis to Nigeria or Uganda or whatever the place it is. And that is how the entire process or the pipeline works. So let me tell you an example. After this procedure is done, so this is just a field trial in Uganda. It's a place called Namalonge. So in that we did a field trial. So you can see how many plants, like almost 3,000 plants have been planted with different entries with the wild type controls and buffer controls. You can see a beautiful plot here. So these are all the transgenic virus resistant cassava that is growing in Uganda. So once that is done, after 12 months after that, then we do the harvest, because the disease that is going to happen is going to be on the storage root. The storage root is buried inside the soil. So after 12 months, we harvest all the so we just harvest all the leaves, and then we take the storage root and slice it and then see. So as you can see in this slide here, this is all the wild type cassava showing the necrosis. So when the farmer sees these type of things, the plant becomes so useless. So people cannot take that. It is not a marketable root. So the farmer goes on a very big loss. So people are suffering, farmers are suffering because of this virus, because it goes and interacts into the storage root, and people cannot eat this. So that is why the kind of technology that we have developed here, the RNAi technology, that you can see this here, the transgenic plant, which has got like for example here the 4-5001 and 1-5001. If you can see, they are all like very clean roots which doesn't have the disease. So it is so marketable, and the farmers are so happy to see these kind of roots because it is completely resistant to the cassava brown streak disease. After that, then what happens is, so we also tested that after second cropping cycle, because like each cropping cycle is like 1 year. So after 1 year, then it goes, then second year it goes. So like you can see after like 2 years also, the wild type plants are showing the necrosis here, and then the transgenic plants are completely resistant here. So to summarize that part of the project is it is highly effective control of the cassava brown streak disease that is demonstrated over the 2 planting cycles. And we have also done the same thing in Kenya. And this is correlated with our own transgenic siRNA expression, that is the small RNA expression. And that is what is happening right now. And after that, like what we are doing is, as I mentioned earlier, we are not only developing plants just in the lab or something, it has to go into the field. So we have to have a clear communication and then some kind of like regulatory work. So there is a lot of work that we do on regulatory. So like the RNAi should not have like any protein product, and the selectable marker that we are using is called NPT2, that is a neomycin phosphotransferase. So it has got a history of safe use in the multiple crops, and there is no wild relatives in Africa. So the gene flow is very minimal. So we make sure that everything is carefully done in Africa, and then we do like this regulatory work for almost 2 years to make sure that that food is safe for people to eat. So we provide that, and then we do the regulatory trials. So as you can see here, these are all the regulatory trials. The first one that I showed was the confined field trials to screen the lines, but now we are doing these regulatory field trials. In that, we will do the yield performance, we will measure all the agronomic characteristics, and we will see the response to target and the non-target diseases. We will see the response to the pest and response to the abiotic stresses, how the plants are doing it, and then also see the stability of the gene, as how it is doing. So when we do all these things, it takes like almost 2 years, and that is what we have done in 2016, 17, and 18 actually. So almost 2 to 3 years we do regulatory field trials in different countries. So once we do that, the next step would be to apply for us submitting the dossier. So we take that, all the documents, and then submit it to the Kenya government, submit it to the Uganda government, and the government will start reviewing all this process. And once that is done, then it will be distributed to the farmers. So we are almost close there. So probably in another 1 or 2 years, so whatever the product that we have developed in the lab, that will be delivered into the farmers' hands, and the farmers will be having these materials in order to go and propagate in their soil. And it is all completely kind of a non-profit organization. So it will go through all the steps, and then the product multiplication and distribution will be happening in Africa. So this is just one example of how we have developed that. And then I'm going to show one more example, like what we have developed here. So we were talking about, I see a hand. I don't know, like, do you want to have questions now, or we can do it later. So, or I can answer now if you have a question, please. Go ahead. Does anyone have a question?
Narayan, continue.
Oh, okay. Okay,
last
good. I saw. Okay, good. Okay. All right. So the second part of my talk is about the biofortification to improve the iron and zinc content inside the storage root. So we talked about the cassava brown streak disease. So that is the disease resistance part. And now we are going to talk about the biofortification part, that is to deliver and a stack product which has the combined resistance to both the disease as well as an elevated micronutrient, that is iron and the zinc content in the farmer preferred Nigerian cassava cultivars. And all the work that I'm talking about is all funded by the Bill and Melinda Gates Foundation. It's a Bill Gates Foundation. So they fund all these research. So as you can see the world map here, it is a mineral micronutrient deficiencies, iron and zinc. As you can see, mostly it is iron deficiency is like distributed mostly in Africa. We have a lot of thing in India also. And then if you see the zinc deficiencies, you can see it is highly distributed here in the Africa. So like some statistics to show that in developing countries, on an average, we have a lot of problem in India too. And like what we are talking about is in Africa, 50% of pregnant women and 40% of the kind of like preschool children are estimated to be anemic. And in Nigeria alone, because Nigeria consumes first the top cassava like food. So in Nigeria alone, 67% of the pregnant women and 76% of the preschool children are anemic. And zinc is also a big problem. So that is why we developed a product for them in order to make sure that these children and these like pregnant women and non-pregnant women, they have to take, because they don't have money to go and take tablets or iron things. So like whatever is their staple food. So we have to provide the nutrition inside the staple food so that people eat that. So that was the main purpose of this like project. So if you start thinking about it, as I mentioned, like more than 800 million people, they consume cassava. It is the second most dietary source of the dietary calories. It has a lot of carbohydrates. So cassava, I know that like in India, in Tamil Nadu, we have all this sabudana and everything, right? We make payasam and other things. So, but that is all coming from the powder, that is all coming from the globules, and it is all about starch. So it provides 80% of required energy per day. It is all about carbohydrates. It has very less amount of nutrients. So in Africa, so like what you see here, all these children and like women, they eat 3 to 4 times of cassava. So like what we have developed in India is mostly, especially in South India, we eat a lot of rice. So like that, you can compare there in Nigeria, people eat a lot of cassava, like morning, lunch, dinner, everything is all starting base is all about cassava. So they make it in a different forms and they eat it. So when they are eating it, they are just having only 6 to 10 parts per million of iron and 7 to 10 parts per million of zinc. So in order to go to a normal level where the children will not be anemic and the pregnant woman needs that amount of iron and zinc, so the target was done from the nutritionists, and they estimated that it has to increase 40 to 50 parts per million. So it has to go at least 4 times higher, whatever that can be done. Of course, the question will come that, like why do we have to go through a biotechnology approach when we can do breeding? Yes, we can do breeding, but there is very less of genetic variation that is happening. So we have tested like almost 1,000 cassava germplasm in South America and African the germplasm, and it is all in the range of 6 to 10. So there is no way that we can increase through, because so that there is very less of genetic variation is happening. So we cannot do that through breeding. So the only way that can be done is through to the biotechnology approach, which I'm going to show in the next slide. So we all know that iron is there in the soil, right? In the soil, we have a lot of iron, but the plants needs to uptake that iron. How does the plant uptake iron? So we have to study as a plant physiologist that like what happens inside the iron biology is, so then like this is the soil rhizosphere outside, and this is the plant cell here. So there is a major transporter. Iron is in the form of a ferric form, Fe3+, and that is what is there in the soil. So the ferric iron, the plant cannot take. So these plants, but the monocot plants can do it, but the dicot plants like cassava, it has a different strategy. It has to reduce the ferric ion to the ferrous ion. So when it does the like this reduction from ferric to ferrous ion through a reductase called as a FRO2, and then it goes into the plant by a transporter, and then it goes into the vacuole or chloroplast or mitochondria, and then it goes and gets stored. So we carefully dissected each and every part of it. So the one that I'm going to show is the success story here, which we have published last year in Nature Biotechnology here. So this is an IRT1, which is a major ion transporter, which takes this ferrous ion like into the plant, and then the ferritin is the one that goes in storage. So we combine IRT1 and the ferritin and make a construct of that, and then send it into the agrobacterium, and that is going into the plant. So again, so these are all the plant genes. So these are nothing coming from any other part. So these are all coming from Arabidopsis. So it is coming from a plant source, and then it goes into the plant. So again, we have done some field trials in Puerto Rico. So because Puerto Rico is quite nearer here, it is the same kind of a hot climate that we can do, and we can kind of like mimic what happens in Africa. So we did in Puerto Rico. So we have done, and this is what is a biofortified like plant looks like. So when you take that ion and then start measuring them into the spectrophotometer, and then you can see that the IRT1 and ferritin field-grown plants. So as I mentioned, the wild-type plants are around 7 to 10 parts per million, and the transgenic plants, as you can see, they are all increased like almost 70 to 80 parts per million. So even some of them shows 94.1. So you can see that there is a tremendous increase of iron inside the plant, and the same plant also does the zinc, because IRT1 is a gene that can transport iron as well as the zinc. So in one goal, we got both iron as well as zinc. So we saw an increase in iron, and then we have the zinc. So these plants are all showing a very good biofortified iron and zinc. And then we also wanted to measure how these plants look like in the field for an agronomic yield. And there is no difference between the wild type and the transgenic plants, and the agronomic yield looks very good, and they are kind of like equivalent to the controls. And then, as I was mentioning, people eat cassava there. So the main staple food that what Nigerian people eat is called as a gari and fufu. So this is a step of like a 4-day step process, like how we ferment for like kind of like idli and dosa. So it is the same way, but it is taking like almost 4 days for them to ferment and then process this. So they make curry and fufu. So like what we did is that we wanted to do the same kind of like process, and then see whether that can be taken inside the stomach. So there is a process called as a bioaccessibility, in which you can take the cassava samples and do a pepsin digestion. So we just exactly mimic the stomach. Pepsin digestion, pancreatic digestion, and then centrifuge it, and then you do ICP analysis. So we did all these kind of experiments. And then when we are seeing that it is not only the plants show more iron, we wanted to see how much iron is like retained inside the storage tank. And then so that has to go inside the stomach. So if we do that, how much iron is retained? So like whatever that we do, it is called 60 to 70% of iron is retained. As I was mentioning, the cassava storage roots are stored in like water for 2 days, and then after that, then we just grind it in a like kind of like a mixie, and then we do a pressing step, and then we do a roasting, and that is how the powder is developed. And this is the base of the cassava, and with that they make soup and they make other kinds of food. So we also did the same thing here, and then we wanted to see how much iron is retained. So as you can see here, so you can see the raw one is this white bars and the curry and fufu. So from the raw to compared to the curry and fufu, you can see 60 to 70% of iron is retained in each and every transgenic line. So when the farmer or when the pregnant woman or when the children is eating this food, like whatever we are feeding, 70% of the iron will go into them. So that is what we have proved here. And then we wanted to see how we do this for the kind of calculation. It is called as an estimated average requirement. So that is nothing but how much nutrients are absorbed, that is divided by the how much of nutrients is kind of like required. So the nutrient absorbed is how much food kind of like a woman or children eat, consume every day, and how much nutrient is retained after the boiling and cooking, and how much nutrient is absorbed. And then the variables contributing to the nutrient required are depending on age, because and also gender. Male eat more, then like women eat lesser, or the children eat lesser. So it all depends on the gender and the age. So when you calculate all these things, we started with a baseline like 5 to 8% EAR, and we wanted to increase like more than 30%. But our transgenic plants, IRT and ferritin transgenic plants, they show like 40 to 50% of iron and 60 to 70% of zinc. So that is a fantastic news, and we are really excited to see that these plants have got, after doing all the calculations, we have increased this iron and zinc in the same plant. And after that, as I mentioned, the first part is the cassava brown streak disease. That has to combine with iron and zinc. So we put like both the pieces together in a single construct, and that goes into the cassava, whereas the Nigerian people are like eating every day. So the cultivars are called these two cultivars. After that, as you can see here, the wild type plants, they show a lot of like disease, and then the transgenic plants are completely resistant of the cassava brown streak disease, and they show an siRNA expression. This is the molecular testing or expression studies that we do by a northern blot. And after that, then we see the phenotype, and these plants also show an increase of iron as well as the zinc in both these two cultivars. So you can see the wild type is around here, and then the wild type is around like 10 parts per million, and then you can see that there is an increase of iron and there is an increase of zinc. So, and then we have also got a field approval in Nigeria. So we have sent all these plants to Africa right now, and that is where we are right now. So we are eagerly looking forward for this field trial and to make sure that these plants are sent there, and then it will be permitted. And I just kind of conclude here to just kind of to thank our like entire team here who has worked from like different crops and different labs, and this is my kind of overall executive team, and this is my lab here. So all of them, they work under me. So they work for my the different parts of tissue culture and molecular work and everything. And then I also want to thank the Gates Foundation for all the funding and everything. And ultimately, like what we wanted to do is to provide cassava, which is nutritious to all the children, which is very important to feed the world. So it is quite a challenge, but then like we are kind of going through this process, and we are very, very optimistic that we can definitely send these products to Africa, where the children and the women will be definitely appreciative, and they can feed this cassava. So I think with that, I think I will just end my presentation, and I would like to have a lot of questions if you guys have, so we can talk and we can discuss more, because that is why I just finished my slides very shortly. And if you have any questions, please feel free to ask. Like maybe I just went fast. Yeah.
First of all, let me thank you, thank the organizers, Dr. Rajarajan and Dr. Narayanan. You have presented so well that now we could understand the importance of zinc and iron, which is very important for the pregnant women, and also especially developing country, and even not even they don't have stable food, but then you are giving a nutrients which is very important for their survival actually. Dr. Narayanan, what is this called in Tamil? Cassava.
Cassava is called like Maravali Kalangu.
Maravali Kalangu. Oh, great.
Yeah.
Great.
So Maravali Kalangu தான் வந்து like we do this all சாபுதானா பண்ணி வந்து like பாயாசம் பண்ணுவோம்ல அதுதான்.
It's like a சீனி கிழங்கு சொல்லுவாங்க, சீனி கிழங்கு. Is it
yes,
same?
சீனி கிழங்கு.
Okay.
இது வந்து கேரளால ரொம்ப ஃபேமஸ். இது கேரளால ரொம்ப சாப்பிடுவாங்க.
Yes, Dr. Narayan, every day morning I'm taking this kelangu without knowing this. I'm taking every day morning. Is it good? Every day morning.
It has lot of starch. So starch carbohydrate is very good. But instead of but it
and the vitamin
have,
A also it seems is very high. Vitamin A and vitamin A.
Yes, it has vitamin A also. It has some of the vitamins, but it doesn't have iron and zinc. That is what we wanted to increase. But it is a very good source of vitamins and carbohydrates.
Thank you, doctor.
I see a question here. So somewhere in the chat, let me get kind of like pull out. Like it just went like that. I didn't know how to do that. Let me see the chat. Or if anyone has a question, please.
Hello, sir. If you permit me, shall I read it out?
Yeah, sure. Yeah.
Yes, sir. There's a question from Debabita Das. Sir, in hills of West Bengal, cassava are widely used. It is mainly in Rajasthan district and Kalimpong. Could you please explain the species they planted? How many species of cassava and how many varieties are introduced in India? Please give information about the wide use of cassava.
Yeah. So I know that also in West Bengal, they eat a lot of cassava. So the one that we are using here, the botanical name is the Manigat esculenta, and that is what we are choosing. And we are choosing that in Africa. I know that in Kerala, there is an institute, and they have developed a lot of agronomical, like kind of like germplasm of cassava. I don't know what is a name exactly what they use there. There should be some local names. So I don't know the local names there, but it is all about the Manigat esculenta was the botanical name of that. But I don't know what local names they are developing there.
Yes, sir. The same person has also asked about the Indian use, sir.
And Indian use, yeah. So like in India, as I was telling, right? So like we use cassava in kinds of different cassava chips or cassava like as sabudana. So people use that because we use as a softened carbohydrate. So we don't like because we already eat a lot of rice. So we don't use cassava as a major source of carbohydrate. But other than that, it can be used as a supplement and also like chips. So that is what people use in like India.
Sir, another
Thank you,
question.
sir. Thank you, sir. Actually, in Darjeeling, people use steamed cassava. Every day they are actually using steamed cassava in Kalimpong market. I have seen during my tenure of service at Darjeeling Government College, postgraduate college. I have seen that type of cassava are used widely in Kalimpong market and in Darjeeling Hill town market, sir. That's why I am telling this one.
Oh, very nice. Oh,
Thank
very
you for
nice. Yeah.
your explanation.
Yeah, sure. You're welcome.
Yes, sir. Another question from students, sir. Sir, can you please tell about the disadvantages of consuming these produced by biotechnology?
Yeah, I'm sure I was expecting this kind of a question for sure. Yeah, so as I was mentioning, all the things that I was so we were developing now, it is all the genes that comes from the plant species and not from any other species. So these are all the plant related genes. These are codon optimized for cassava, and then like before it is like it will be released into the public, like it will go through a lot of testing of regulatory testing that is happening now, and after that then it will happen in testing in kind of rabbits and other like species and mice and other testing, and then it will go through the human testing for palatability for tasting. So by the time it will reach people, it will be another like almost 4 to 5 years. So we have carefully developed all the toxicology tests and the regulatory test from each and every country, and that is when it has been developed. But in America, most of the products are genetically modified, but like in Africa, these are the couple of first products that we are developing right now. But the way that we are doing now is there is no other way that we can do it in breeding. So we are always preferring breeding as a first method. If we don't have a process in which we cannot do without breeding, then only we go into the biotechnology approach so that it can go. But right now, I'm sure that you must have all be aware of the gene editing technology. We are also developing a lot of tools with the gene editing, and that is not exactly like the genetic transformation. It is just a small snip in the gene. So we are also doing gene editing of that.
The question from Madan. He has asked about the climatic conditions for growing cassava.
Climatic, it has to be hot. It has to be hot, hotter, hottest. So that is what cassava like. So that is why we are growing in India. Like as you see in Kerala and everything, it grows in Africa. So most places, like it needs, it is a crop. It is not a very demanding crop. It can, so like even it can resist in drought. Even if it doesn't rain, it can resist like almost 2 to 3 years. So that is why it's called as the poor man's crop. And so like it can be grown in hot and humid conditions.
Actually, the same person, sir, Mr. Debrata Das, has again asked some information, sir, because the person is in need of those person for the transgenic stock for mycorrhizal research. So yes, he's requesting.
Can you please repeat it? Can you please repeat the question?
Yes, the person has requested for an resource person's email so that he wants a transgenic stock for mycorrhizal research.
Oh, for the mycorrhizal, so
Yes.
mycorrhizal interaction. Okay, I can find out like what other sources, and maybe I can just kind of like email the Dr. Rajarajan, and then maybe he can contact him.
Yes, thank you. Any questions from audience? Okay, so I assume that for the wonderful talk, and now I request our Dr. R. Rajarajan sir to deliver vote of thanks.
Yeah, thank you, Anand. A very good morning to one and all. Department of Botany extends their gratitude to the Secretary, Principal, and the management for permitting us to conduct the one-day international webinar. I would like to thank the resource person and my dear friend, Dr. Narayanan, for his valuable, wonderful, and inspirational address on Cassava Biotechnology. I would like to thank the head, Dr. Muralidharan sir, and other faculty members of our department for their support in all the ways and means. I would like to thank the participants all over the globe, and last, I would like to thank our dear students of our own department for their cooperation and patience. Thank you.
Thank you, sir. Today's webinar was really informative, and I surely believe that audience would have gained knowledge from today's session. Thank you, one and all.
Thank you.
Thank you, sir.
Yeah, thank you.
Thank you very much, Dr. Narayanan.
Thank you.
Thank you,
Thanks,
Dr.
Rajarajan.
Narayanan. Thank you, Dr. Rajashekharan. Thank you.
Thank you, thank you.
Okay, thank you.
Bye, sir.
So shall I leave? Is that okay?
Yes. Yes, yes.
Yeah.
Certainly.
Yes, sir. Yes, sir.
Yeah, sure. Thank you. Okay, bye-bye.
Bye, sir.
Bye.
Sir, can we leave the meeting, sir?